really interesting thing about bacteria and archaea is that they have very fluid genomes. And so when we talk about genomes, we're just referring to the genetic material of an organism. And so for cellular organisms, of course, this means double-stranded DNA, but remember, that's not the case for viruses. And so while they share the same genetic component, there are a lot of differences in the way genomes evolve and kind of behave between prokaryotic and eukaryotic cells. And this is so different that when we talk about prokaryotes, we need to use special terminology for when we're referring to the genome. So there's this concept called the core-pan concept, and what that is referring to is basically in what proportion or basically how common are those genes among a species or a population. And so when we're talking about eukaryotic genomes, we know that for the most part, the human genome, for example, is pretty consistent. We were able to map it, we know what genes are present, and while there are a lot of variations within those genes, the genome itself is pretty stable. With prokaryotes, however, they're not stable. There's a lot of fluidity, and things will change between different members, even of the same species. And so when we're looking at those genomes, we define the core genome as those genes that are present in all members that you're looking at. So I'm just going to pretend we're talking about a species, let's say E. coli. So the core genome would be the genes that are found in every member of E. coli. And so this is not actually all genes or even a large majority of the genes. It's believed that the core genome of some bacterial species can be less than half of the genome, in fact. And so all of those other genes that are not present in all of the members are called the accessory genes. Sometimes these are also called variable or dispensable, but those genes will make up the accessory genome. And so you can see in these Venn diagrams, you have the core genome, which is shared by all members. You have the accessory genome, which is shared by maybe some, maybe only an individual member. It kind of depends. And these things taken together make up the pangenome. And so that's the full complement of genes in a given species or population. So it's the accessory plus the core genome. And so this means basically all of the things that could possibly be in members of that species or population. And so the ways that this fluidity kind of takes place is primarily through mutation, so changes in the DNA sequence that are then passed down to offspring through what's called vertical gene transfer or vertical evolution, as well as horizontal gene transfer. So in horizontal gene transfer, this major mechanism of genome evolution, you have genes passing from one organism to another, but not in the parent to offspring motif. So these can be neighboring cells of closely related taxa or of distantly related taxa, depending on the situation. And so with genomes, basically mutations can contribute to new functions as well as horizontal gene transfer because mutations can develop pretty quickly. And this is especially common if there's some sort of duplication event that basically creates a spare copy. So say that these genes were essential function, if there were a lot of mutations that developed in these essential functioning genes, the organism would be unlikely to survive. However, if that genome was duplicated, you had an extra copy of it, these genes could kind of stay stable while these other spare copy genes can develop new functions. And so that's the neofunctionalization shown here. So that's one of the ways that mutation can really contribute to new functionality in prokaryotic genomes. Of course, with horizontal gene transfer, rather than having to wait for things to kind of develop over time, what will happen is you'll basically get some sort of like pre-packaged set of genes from your neighboring cell. So these are very powerful, but very different mechanisms for how new functions are gained. In addition to all these new functionalities and gaining things in your genome, there is also the ability of prokaryotes to shed non-essential things. So this is called genome reduction, and it's very common in prokaryotic cells, which is one of the reasons that you can see here, the number of protein coding genes and the genome size is very closely related for prokaryotes, as well as viruses to a lesser extent. Once you start getting up into eukaryotes, they have a lot more non-coding genes or non-coding regions of DNA that don't directly relate to the encoding of a protein. So with bacteria and archaea, what they'll do is basically shed genes that aren't essential. And so this is very advantageous when you're thinking about like a single cell and the energy it takes to replicate a genome. Shedding things that aren't adding to your cell can be a very powerful tool. And so all of this is to address the fluidity of prokaryotic genomes. And I love this quote by Ed DeLong, who's a marine microbial ecologist who I really appreciate a lot of his work. But he has this quote that it turns out that a genome is a verb, not a noun, a process, not a product. And so there are all these things that are influencing the organisms and the things that make up the genome. And so all of this plays a large role. But what we're going to focus on in this section are the mutations and the horizontal gene transfer and as the major factors that contribute to what a prokaryotic genome is when we consider it as a static entity.